All right. Time for another episode of AP Biology Review. Last time we were talking about, we've ended with movement of materials through the membrane, talking about that phospholipid membrane, cellular membrane. So I'm going to kind of continue in the same vein and start with just maybe I could touch a little bit on this other one, so it could be a review of review. But anyway, let's get going. Facilitated diffusion is one aspect, an important aspect. This is transportation with help to facilitate. This is diffusion, so it's a passive transportation, so it's always going to be from high to low. And one issue that molecules and substances have is getting across this membrane. Remember, it's that hydrophobic area, and they need to get across it. The best, most efficient way are through these kind of carrier proteins, tunnel proteins, ion gated channels, all sorts of things on the membrane that are interspersed. They're talked about integrins spanning the entire membrane. These channels are going to be specific to the different types of molecules, and they will let them in, going again high to low, passive diffusion. And then so there's some pictures here of these kind of carrier proteins, and I have a little nice, found this online for it. Picture didn't come out quite as well. But that's facilitated diffusion. Remember, there's all sorts of things on there. If it was going from high to low, remember, that would be active and ATP would be required. On osmosis, didn't spend a whole lot of time on osmosis in the last part. Osmosis, diffusion of water, and we have the different terms associated with osmosis and how water is going to flow. You have the hypotonic solution, the isotonic solution, the hypertonic solution. I am skeptical that the College Board will ask something as easy as this, since you just type in hypotonic solution into your Google machine and you get the answer. But anyway, a hypotonic solution, we're going to have water entering the cell. All right, it's going to swell, and hypo means less. So we're going to have less solute outside of the cell, which means I have a higher concentration of water outside the cell compared to the inside, and water will flow in. Isotonic solutions are the same, equal amounts of water on either side of the membrane. You're going to have equal amounts leave and enter. And then the hypertonic solution is where we have the water flow out, because, again, we have more solute outside of the cell. So like solute being what's dissolved, and the water wants to balance it out, so you have equal concentration, so water will flow out of the cell. Again, just a recap of these terms. And I believe I'm done with transportation. Nope, water potential. That's right, water potential. I had one question last week that had you calculate. It's pretty easy, but some of the calculations, it's possible to throw a water potential question at you. And it's passive. We're dealing with a passive situation, and it's always going to be a high potential to a low potential. Right, the area may change, very much similar to the osmosis, and water is going to move from a area of higher concentration to area of lower concentration. It's really the same idea, but we're talking about a potential now instead of a concentration. You have your formula here. The water potential is the solute potential plus the pressure potential. Remember, open beakers, the pressure potential is 0. If you have a question and they don't give you the pressure potential, it is 99.9% likely that that pressure potential will be 0. What you will have to do is most likely associate with psi s, the solute potential. That equals minus I CRT, the ionization constant, I, 1 for covalent compounds, 2 for ionic. And remember that sucrose, or sugar, is 1, because it does not ionize with water. And then those ionic compounds, it's usually salt. It's going to be NaCl, 1 for Na, 1 for Cl. It can be more complicated than this, but I don't see it getting more complicated than a 1 or a 2. Your concentration is just what it is in molarity. Usually that's given. R is going to be your pressure constant, which again should be given. And then the temperature, which should be given. You'll probably have to add it to Celsius to 273 to get Kelvin to get the right answer. But otherwise, it's pretty straightforward. And then you would calculate your water potentials. You have hopefully 2, and you can compare, and the water will move into the area of we'll move to the lower potential, high to low. That's water potential. Not too bad in the calculations. All right, so now we're going to start Unit 3. Unit 3 deals with a number of different things. The first one is enzymes, which I would have put in Unit 2, but that's just me. And then cellular energetics, respiration, and photosynthesis we'll deal with the next video, but not today. So enzymes are these these are proteins that are catalyzing reactions. They are lowering the activation energy of a reaction and causing them to happen more quickly. Without enzymes, some reactions wouldn't happen or would take hundreds, if not thousands of years to accomplish. So your basic structure of an enzyme, you have the enzyme and the active site. The active site is a particular spot and shape of the protein. Remember, these proteins is two-dimensional, one is a three-dimensional shape. And if the substrate is the molecule will be acted on in the enzyme. If there is any kind of deviation here, the substrate can't fit, the enzyme is not going to work for that particular molecule. So what's going to happen, the substrate is going to attach, it's going to bind to the active site. Once it binds to the active site, that's when catalysis happens, and at the end you have whatever it's supposed to do, whether it's bringing molecules together and forming bonds, whether it's breaking them, as in this case, many different things, but that's the active site. It's the important, right? That's the shape, and it's determined by the amino acid primary structure and secondary structure. It all goes back to the shape of those proteins and how they interact. Any kind of mistake in the folding or anything can affect the active site, which then would cause this not to work potentially. And then I want to point out that temperature is heavily involved in enzymes since they're proteins. Proteins will denature at a certain temperature. So if you may see a graph of here we have a rate of reaction versus temperature. And you have your optimal temperature, and then you get to a certain point and your rate goes down to 0 because the enzyme is denatured. Alright, so it's a couple notes here. Enzymes are proteins, so they go back to the genetics, the DNA. Mutate the DNA, you can change that active site, which then causes the protein to be non-functional most likely. In rare situations, and I'd like to know when they can become better. And I did talk about denaturing. And then lowering activation energy is how they work. Outside of cell, these are, if you look at two different enzymes here, you got A and B and A and B. Enzymes are all different, so these two enzymes will have different optimal temperatures. They're going to denature at different temperatures. And then also remember pH is also a factor of this. Some enzymes work best in acidic conditions, some work best at basic conditions. I guess that's slightly basic, this is a little skewed. And most are going to work right around neutral. But keep that in mind, you might see something like that. Alright, enzyme regulation, we're almost done. There are different things that can happen to regulate, and I put this in quotes because sometimes, toxins or whatnot, poisons can mess up enzymes as well. But here you have your regular reaction substrate attaches to the active site, and you have the reaction happen. We have competitive inhibition, right? We have something that is not the regular substrate that fits into the active site, blocking the substrate, right? So this doesn't happen because this particular inhibitor is blocking the active site. This could be intentional by a living system, also to stop production of something. Then you have non-competitive inhibitors, also called allosteric regulation for you nerds out there. This is where we have an inhibitor attached to a different spot on the enzyme. Remember, enzymes are proteins and they're very sensitive to any kind of change. This will interact with the R groups of the enzyme, and as a consequence is going to change the shape of the enzyme. That change in shape of the enzyme will then cause the substrate, again, not to be able to attach properly, and therefore not work. So non-competitive, a site other than the active site, blocking, causing a change in shape which blocks the substrate from attaching. And then we have another, so I like this picture because it also included feedback inhibition. Right, so if you look back to A, this little green thing was a product. Right, sometimes systems, because you don't want to waste resources, so you don't want to make too much if you don't need it. Sometimes you have a product will actually come back and bind to the enzyme that is making it, causing it to change shape, which again disallows the substrate from attaching to the enzyme. And this is a non-competitive inhibitor, although they can also, I suppose, be competitive inhibitors as well. But you'll see that too, feedback inhibition. And that's enzymes. I think there could be a lot of questions about enzymes, really graphing things and asking questions. I think that's it. Yep, that's the end of my show. All right, well, enjoy, and good luck with your questions today. Subscribe. I'm still like 10,000 subscribers short of monetizing my channel. Maybe one day. See you.